we're going to do that today. And so now this is the last group of plants to talk about. This is the group of plants, of course, that have the most, they're center stage when it comes to commercial crop production, whether you're thinking about food or medicinal properties or fiber or fuel of all kinds. So it's a big group to learn about. And so now we're on the last line of the phylogenetic tree, looking at the other group of seed plants. Okay, so we already looked at the gymnosperms, but now we're going to be getting into the angiosperms. And so huge difference in the sense that now we have flowers and fruits to think about. The gymnosperms had cones and then eventually seed production. With angiosperms, we're looking at flowers and then fruit and seed production. So they are encased in some kind of fruit tissue. Here you're looking at an orange, so that's called a fleshy fruit. We're going to find that some fruits are dry. They call them dry, like beans, beans in a pod. That's actually a fruit, even though your parents might have told you to eat your vegetables and eat your beans, but they're technically fruits. Corn on the cob is a fruit. So we'll get into some of that. First thing I'd like to get you thinking about is the flower. So we're going to be zooming in quite a bit to what's happening inside the flower, and in particular, a couple of key parts. So as far as flower anatomy, I think I mentioned this already, but I'll go through it quickly. Please know the term sepal, which is when the flower bud hasn't opened yet, the sepal is covering the flower bud and protecting it, usually green. Petals are usually showy if it's a flower that is destined to be pollinated by another organism, it'll be showy. And so we're kind of working our way from the outside of the flower to the inside. So the sepal, then the petals, and then the male part of the flower, if it's there, will be next in the pattern here. And so taken together, this anther and the filament, if you include both of those, you call it a stamen. So the male parts of the flower are all listed here. The way I remember it is anther is close to the word antler, and I think about male deers having antlers. So that's how I remember anther is part of the male. So that's actually where we're going to find pollen being formed. The filament is actually just the stalk that holds the anther up. The way I remember that is filament on a fishing line. My dad used to like to take me fishing. So I think about the filament fishing line, male, my dad. So that's how I remember that. Or MEN is in the word, men, filament, that helps. Stamen, MEN is in the word again. And as part of the male part of the flower. And then center stage is the female, right in the center. And so all taken together, you can call it the pistil or the carpel. Either term I see quite frequently still. I tend to remember pistil because my dad used to say, when he'd get a little irritated with me, he'd say, "Oh, you're such a pistil." And I remember that. So that's how I remember that. And then stigma, he would also say that females are enigmas, which rhymes with stigma. So I remember that. So the stigma is the top part. Style, because women have style. So the style is the star part. And then the ovary, of course. Females have ovary. So I'm being a little silly with all this, but that's actually how I remember a lot of stuff. I connect it to something personal and it sticks. And it's a tried and true way to learn stuff. And it makes me smile because it makes me think of my dad in this case. So he's no longer with us. So it makes me smile. All right. So we have stigma, style, and ovary. And then inside the ovary, you may have one ovule or you may have many ovules. It depends on the plant species. Here you're seeing 6 in this particular case. So then when you zoom into the ovule, eventually we'll start talking about an egg in there, okay? So we got lots of parts here to think about. Please be prepared to either label a picture or pick out the definition. If, given a definition, you know the term. Okay, there's another one here too, just if you prefer to look at this one, I just put this in for studying purposes, if it helps at all. Sometimes you see these words, androecium for the male part, gynoecium for the female part. So those are some older terminology, but That's why I put this in here, because sometimes you'll see that, especially in lab, you might see some of the slides or something like that labeled that way. Okay, and then you were supposed to look at lily. Last week, we apparently, the stores around here didn't have lily flowers for sale. It was very challenging to find any. So that was a weird situation. Um, but again, the reason we pick lily is that the parts of the flower are usually very big and easy to see with the naked eye, and the ovary is very obvious and very swollen at the base, so it's really easy to see. So we're going to be zooming in first to the female part, which is the center portion of most flowers. And so the mega or female gametophyte is what we're thinking about here. And in the angiosperm realm, often they replace the word megagametophyte with embryo sac. And they just do. I don't know who started that. I don't know why, but they do. And so you'll often see it labeled as an embryo sac, but it's really the female or the megagametophyte that we're thinking about. So some similarities to the gymnosperms, we still see that there's two different kinds of spores. They're heterospores. There's megaspores and microspores. So we already saw that with the gymnosperms. The megaspores will stay. They're not going to get released. So they stay in the sporangium tissue, the megasporangium tissue, and eventually the megaspores will grow in to become the megagametophyte or the embryo sac. The microspores, they stay inside the microsporangium. They do not get released from the plant, and those will develop into the microgametophyte, which are pollen grains. Okay, so this is similar to what we saw with the gymnosperms, except, we're dealing with flowers now instead of cone tissue. So now you've seen this next slide, so you don't have to write all this down necessarily, but you've seen this with the gymnosperms. So the spores remain on, they don't get released like they did in the bryophytes and the the plants. They stay on, they get well protected, they're in the megasporangium or the microsporangium, and there's no free water necessary, so the plant doesn't need to wait for a watery day to deal with fertilization. And again, like the gymnosperms, the angiosperms, they have this alternation of generations again. And the gametophyte is going to be held on and developed in protective tissue of the sporophyte. So the sporophyte is really dominating. It's the plant that you see. The gametophytes are the pollen grains and the embryo sac or the megagametophyte inside the flower, deep inside the flower. And again, it's the seed that will get released. Just like what we saw with the gymnosperm, it's the seed that gets released out from the sporophyte. Okay, now I'm going to get into a little bit more detail than what we did with the gymnosperms when we think about reproduction in the angiosperm. But in many ways, the angiosperm reproduction is more straightforward than what we see in gymnosperms. So I'm giving you the better version. It's a little easier to work through this. So in the angiosperms, what we see is that the megaspores that are held within the megasporangium, they put all their eggs in one basket. They reduce all their megaspores to just one, which to me seems a little odd that they count on that one megaspore, but they do. So the megaspores, instead of having lots of them, it's just reduced to one, just one. And so they give that a special name. They call it the megaspore mother cell. So it has a full complement of chromosomes. So we say it's diploid or 2N. And so its job is to undergo meiosis. So the megaspore mother cell goes through meiosis. When you do that, if you have one cell going through meiosis, you end up with 4 cells at the end of meiosis. And they're all haploid. They have half the number of chromosomes now compared to that megaspore mother cell. What we're going to see is that 3 of these die off and only 1 remains. That one that's remaining is called the functional megaspore. Okay, so that megaspore mother cell, there's just one lone megaspore mother cell. Here's a picture. where what we're looking at are some ovules. You can see two ovules here, one on the top and one at the bottom. There's some others on the other side that didn't make it into the picture. So what you're seeing here is this kind of pink, grainy area. There is a cell that's starting to form there called the megaspore mother cell, right? And you can see that it's surrounded by, I'll show you a better picture, but it's surrounded by sporophyte tissue that's called integument tissue. If you're and looking at this entire thing, like this whole structure right here, that whole thing is called the ovule, right? So it's this megaspore mother cell, just one. Okay, this is megasporangium tissue that we're zooming in on here. And instead of having a lot of megaspores in there, there's just one. So now that megaspore mother cell has to go through meiosis. And then those 4 cells get made. So that's what's being shown here. So here's those 4 cells that got made through the process of meiosis. And then we say that 3 die off, and you're left with 1. So that remaining one is called the functional megaspore, and it has half the number of chromosomes. And you can see it's still held within the megasporangium, not released. None of that was released. It's all protected. Here you can clearly see the integument tissue surrounding that, so that it's just beefed up protection here. You have the megaspores retained within the megasporangium surrounded by the integument to further protect it. And then you have this tiny little opening that's called the micropyle. I would like you to know that opening name, micropyle. And then so it's just that little section that's open, and then the rest is surrounded by integument tissue, and then if you work your way in, then the megasporangium and then the megaspore. Okay, so now that functional megaspore has a job to do. It needs to divide through mitosis. So it's just going to, duplicate itself essentially. So it's going to do that 3 times. It's going to go through 3 rounds of mitosis. So that's what you're seeing here. So when that one cell divides, you form 2 cells from that, and then you do 2 more rounds. Well, 3 times of mitosis essentially. So what's weird though is that you end up with 8 nuclei, which makes sense. If you go through 3 rounds of mitosis, you should end up with 8 nuclei, but you only end up with 7 cells. It's weird. What they do is they tuck 2 nuclei into 1 cell. And then there's the other cells get just 1 nucleus each. Okay, so what you see here at the bottom is this polar nuclei that they call it, where they're pointing to 2 nuclei in the center. That's why they call it polar, because they're usually sitting in the center. That is 1 big cell there. And so you have 2 nuclei in that 1 big polar cell. And then these other cells that you see here each have their own nucleus. So let me blow that up a little bit, see if I can get that. All right, so these polar nuclei, I'm trying to show that. I don't know if you're sitting or not, but it shows up a bit better. And then to these, there's 3 cells here. So that is the megagametocyte, female or the embryos. So they say, oh, look at the embryo stuff. This whole thing. So that's all there is to it, which is much simpler than if I had told you all about Angiosperm. They go crazy with how many cells. It's very small. But anyway, so what I want you to know is that where the micropyle is opening, that's where they put the egg. The egg is usually right there, which makes sense. The sperm's got to get it's going to have it right by the egg right there. And then it's surrounded by 2 synergies. I'm still not quite sure what those synergies do, but they do see that they're involved in somehow they facilitate the sperm and egg fusion. And they don't know exactly why or how. But when they've done some experiments, if they get rid of the synergy, they see the fertilization rates go down. So they think that somehow we get the sperm and egg to. Then we have the cell here. And then these three at the far end, they're called antipodal cells. What do you know that? Antipodal cells, antipodal, so when we zoom into the megagametophite here, some of the key things happening are the egg, of course, and then we also have second cell there is the polar nuclei. That becomes very important as well. So hold on to that thought. Under the microscope for real, this is lily. So it's very common to look at how this happens in lily flowers because their ovaries are so big, it's easy to make slide preps of. It's just easier to see when you look at it. But lilies, so when we look at this, I'm not going to get into details here. But in general, I see how this whole area here is possible. Then there's this becoming the microfile is actually pulled up. Once the sperm gets here, the microfile fused over that opening. So you'll get that happening. Actually, it's at the other end. Sorry. The micropyle. Anyways, so when we look at this, here's one that's labeled, and you can see it's not perfect. Like the drawing I showed you, everything was just perfectly, set in the megagametophite, but in reality, it's a little messier than that. So we won't be asking you to identify something like this, but if I give you a perfect drawing, then I would ask you to identify what's what. So if we go back to, for example, a drawing that looks very similar to this. Okay, if I were to ask you to label something, it would be perfect, not a messy microscope picture. But they're fun to look at and try to figure out what's going on. And so the integument tissue around eventually will come through there. Okay, here's a better, like a more idealistic drawing of it. So antipodals, central cell with a polar nuclei, the egg, synergids, and the micropyle. Whoops. And then the integument surrounding all of that. Okay, so now the way this is set up is good to go. The egg is there. It's like, okay, my job is done now. It's time for the pollen to get there and the sperm to get delivered. And so we don't need free water for this. And actually, whoever made this slide, whoever prepped this slide should get a gold star because this slide is showing double fertilization going on. So what we're going to learn, here's the egg cell right there. And that's one of the sperm right there. And it's just about to fuse with the egg. Here's another sperm coming along and fusing with the polar nuclei. So we're going to find that in angiosperms, they're going to deliver two sperm, just like what we saw with the gymnosperms. Their pollen delivers two sperm. But with gymnosperms, only one pollen makes it to the end. The other one dies off. Angiosperms, they use both. Both sperm do something important. One sperm will fertilize the egg, super important, because that's how you get your baby sporophyte. the other sperm is going to fuse with these polar nuclei. And that's important because then that cell will develop into the endosperm, which feeds the baby. That endosperm is part of the seed and super critical because it is the source of nutrition. So really, really, both sperm have a very important job to play. So if that egg gets fertilized, this is a very young embryo. What you're seeing there, that little red blotch, that's like just a handful of cells now that have grown from that fertilized egg. And so this is really young. This is very soon after fertilization. A bit bigger picture. So that's going to keep growing through just mitosis, keep growing and differentiating into the embryo or the baby sporophyte. So what I just described to you is called polygonum, this top row. Polygonum is what I just showed you. Most, 70% of flowers that they've examined will do this polygonum type megagametophyte or embryo sac development. And then you can see that the remaining 30% of flowers can vary on how they do it. So I only want you to know the most common. So understand that the word is polygonum, and then what we just worked through is how this works, where you end up with 8 nuclei and 7 cells by the time the megagametophyte or embryo sac is ready. Okay. But you can see that Lilium makes the list here as doing something different, right? So they have a funky method of fusing cells partway through this process. And so they end up with multiple nuclei in cells. So really kind of different. But like I said, the reason we pick Lily to work with in the lab is because their flower parts are so big, it's easy to see what's going on under the microscope. So it just depends on what the other 30% do. So the polygonum type has 8 nuclei, but you can see that it can range anywhere from 4 to 16 nuclei or in cells. And then the endosperm. So with the polygonum type development, if you have 2 nuclei in that polar or central cell, and then a sperm nucleus gets added to it, that's 3 nuclei. So that's called triploid. Is that on there? No. So the endosperm and polygonum. Yeah. So the endosperm is being developed from the central cell that has the polar nuclei, those 2 nuclei, right? So there's 2. And then the sperm nucleus, going to add that one. So that's going to, that second sperm is going to fuse with this central cell. So now you're up to 3. And so we call that 3N or triploid. So endosperm tissue actually triploid, which is, like most of our cells are diploid. So this is a little funky that you have triploid. And then you can see that depending on what species you're looking at, it could even be pentaploid 5N. I don't know what 9N is called. I know there's another term for that, but I don't remember. 15N. So that's just the number of nuclei showing up in those cells. It's crazy. It's really weird. Especially when you think about animals, how most of our body is 2N, diploid. So they do it differently in some regards, for sure. Okay. This is just a blow up looking at that. Okay, so what do we got here? Questions on the megagametophyte process. So 3 rounds of mitosis, 8 nuclei, 7 cells, knowing that there's an egg surrounded by 2 synergids, and then the other 3 migrate to the other end, and those are antipodal, and then those 2 central nuclei are stuck into one cell, the polar or central cell. That's all there is to it. That's pretty straightforward compared, especially when you look, you didn't learn about how gymnosperms do this, but it's quite a bit different, more complicated. So this is the embryo sac or the megagametophyte. And so once you have the egg and the whole embryo sac mature, ready to go, the egg is ready to go, then it's got to wait for the sperm to get there. So now we have to think about the male gametophyte or the microgametophyte, and that's really the pollen grains that we're thinking about. And so now we are going into where the anther of the flower is, and the anther is going to house the microspores. So we have to think about that. You're going to have this, remember that all the microspores get retained, they don't get shed out. And so they're still stuck inside the microsporangium, which is the anther part of the flower. It's right there. And so we have a similar terminology. They decided to call those microspores microspore mother cells, and you'll often see it abbreviated as MMC. So you have these microspore mother cells, lots of them, not just one, like what we saw with the female. There's lots of microspores, lots and lots. And so each one of those is going to go through meiosis, and if each one of those cells goes through meiosis, you end up, for one cell undergoing meiosis, you have 4 progeny or daughter cells, but they have half the number of chromosomes. And then each one of those will develop into a pollen grain. So you don't have 3 dying off like what we saw with the female version. Everything, kind of think about, how there's lots of sperm produced in animals and much fewer eggs. It's kind of the same analogy here, where every single one of these haploid cells can develop into a pollen grain. Lots and lots and lots. And so then each of those pollen grains will consist of 2 cells. One of those cells is called a generative cell. And initially it's just called the generative cell because it hasn't divided yet to produce sperm, but it will eventually. It just doesn't do it right away. And then the other cell is called the tube cell, and that's the cell that elongates out and grows out of the pollen grain. And it's like the tunnel that the sperm will migrate down so that they can get help to reach the egg. So the pollen tube is really to deliver the sperm. So when the pollen grain gets shed from the flower, there's no sperm there yet. They're not there yet. The generative cell is there, but that hasn't gone through cell division to produce the sperm. So they say it's immature. The microgametophyte is immature when it's shed initially. Okay. So we're zooming into the anther. And when you look under the microscope, each anther has 4, like they call them chambers, right here. Okay, so there's 4 chambers. So this is where we're zooming into the microsporangium tissue. Okay, so there's like 4 microsporangium regions, I guess. And then inside are the microspores, which go through that meiosis, and then they start developing into pollen grains. And if you look at them under the microscope, this is actually a flower that was cut in cross section. So the central part of the flower is right off in this upper right here. That's the female part of the flower. So that's the ovary that they cut through. You can see that it's like 6 ovules in this one. And then the ovary tissue. So then the male part is surrounding that. So you're seeing only one complete anther, but you can see that there's many that must go around all the way. They just didn't take the whole thing when they created this image. So these anthers, you can see there's 4 chambers, 4 sections. So This is microsporangium tissue, and those darker red, those are microspores. Okay, so all of those have to go through meiosis, and each cell will produce 4, and then each of those will develop into a pollen grain. So that's being shown here on the schematic, where you have the megaspore or microspore mother cell going through meiosis that produces 4 haploid microspores, and then they're trying to show that each one of those will develop into this pollen grain, which has the generative cell and the tube cell. And then eventually that generative cell divides to produce sperm. But that doesn't happen right away. And then the pollen grain is very tough. It has a coating or a wall that is really resistant to adverse weather conditions, and it's amazingly persistent. You know, pollen can stick around in the environment for a long, long time. So the pollen grain is the microgametophyte. Here, this is a more mature situation now where you do see pollen grains now. So each one of those pollen grains is the microgametophyte. But there's no sperm there yet. That doesn't show up yet. It's not really until you have pollination. So once the pollen lands on the stigma of the flower, and if everything's a go, the pollen tube will start to germinate down. The female part has to go through the stigma, the style, and make it all the way to the ovary. And as that's happening, the generative cell will divide to produce 2 sperm. And then the sperm will travel through the pollen tube and hopefully make it to the megagametophyte or the embryo sac. So you can see it's coming out here. It's 1, 2, 3 pores. And so it's coming out of the pollen tube's coming out of one of the pores there, the opening. Um, 3 openings. Is that a monocot or dicot? Do you remember? Yeah, that's a dicot. So it's not until the pollen grain lands on the stigma, and then they know that there has to be some kind of, they think it's chemical, I guess communication might be a way to say it, where the stigma and the pollen grain have to match, so to speak. And so getting that pollen grain to start to germinate out the pollen tube and get the sperm to divide, you know, there has to be this kind of acceptance of the stigma. They think it's controlled by the stigma, that the stigma is either allowing the pollen grain to go ahead and germinate or not. And so that's a very active area of research. I haven't kept up on the reading on that too much, but I know that they've been trying to figure that out, because you can pollinate all you want, and sometimes the stigma, it just won't happen. The pollen grains just sit there and they won't germinate out the pollen tube. The sperm won't get produced. And so they say they're incompatible. And they're trying to figure that all out, of course, because if you can't get pollination to happen, you can't get fruit production. And obviously, we want to grow plants that provide fruit in so many of our crops. Here's an aperture, just one. So this is a monocot idea, just one. Very rather smooth pollen grain. Here's one that's more wrinkled. This one has 3. So pollen grains are amazingly diverse in their morphology. They can be really fun to look at under the microscope. The diversity is stunning. They say every plant species has a unique design, and that's why it's so useful to use in forensics. I think I may have mentioned they've used pollen to solve crime, murders. They can tie the murderer to the scene of the crime through the pollen grains that they collected either on the person or in the vehicle. So that's how well preserved they are. They don't break down, and they're so unique. Really quite beautiful. That one, I'm used to thinking pollen grains are pretty circular, but or spherical. That was interesting. Also, it's like a boomerang, that kind of thing. This one, the one on the bottom left, I can't even see the apertures. So I don't know what that is. If it's monocut or dicut. That was a tough one. But basically, what we're thinking about is that the pollen has to make it from the anther, either from the same flower or a different flower, and get to the stigma. All right, so that's the definition of pollination in the angiosperm world. It's the transfer of pollen from the anther to the stigma. Here you can see several grains, pollen grains have landed, and they're in different stages of getting the pollen to growing. And so they have to, essentially, they say it digests its way through the stigma and style, and then the two sperm get discharged into the megagametophyte there. And one of the sperm goes to the egg, which you've got to get that egg fertilized, and then the other sperm goes to the two polar nuclei. And so they call that double fertilization. That's unique to the angiosperm. Angiosperm do not do this. Double fertilization. So that term is, it's kind of a weird term, but what they're referring to the egg getting fertilized, polar nuclei, and the second sperm getting antipolar. The antipoles? Don't know. I don't know if anyone knows, to be honest. Someone must know at least an inkling of what they do. But I mean, they do develop after this point. There's more. Okay, the latest hypothesis I heard that these antipoles after this double fertilization, so the egg develops baby spore. This develops last the nutrition from those kind of gets sucked into developing. I don't know if that's how the experiments that they've done, but they do have those antipodal cells, and like even with the non-polygonum type, they have some version of that going on, where you have those antipodal cells in pretty much every case. So, but exactly what they do, I don't know. They don't know. So there, I put in a couple of slides here that have the entire life cycle put together on one image. And then I've separated them out too, if you want to see them that way when you're studying. And then there's another image too. So this is pretty much everything we just talked about, but there's some visual kind of a flow chart idea. And they have both the male and the female here. Looking at the different parts of the flower and what's happening with each. And then they're separating it out. You know, they're showing before meiosis is on the left, so you can see 2N, that's diploid, and then with the meiosis event off to the right. They tried to color code it as well, so you can see what's happening there. So this is a good resource to use as you're trying to work through this process. The whole embryo sac development coming off the megagametophyte. I think spend some time with this one, or if you prefer, there's another image in here too. I put a couple in just so you can. There's this one as well. It's not as explanatory. I think the one we were just looking at, they did a little bit more explanation of what was happening. So this one's a little bit more abbreviated. But it, you know, sometimes it just depends how your brain works, how you like to imagine or visualize what's going on here. So whatever one you find useful. But I will be asking, this is the only life cycle I'll be asking any details on terms of step by step. I haven't done that with any of the other plant groups yet. So, but the angiosperms, moving forward through the rest of the class now, we're going to be really zooming in on angiosperms. So I think it's appropriate that you have a good understanding. So we've only made it to the point where you got the sperm there and you get this double fertilization, but then we have to talk about what happens after that. That's not the end. So when you think about here, they're showing the megagametophite. Not ready to fertilize. So once sperm is back up to the and then technically the embryo will be the baby sporophyte. And then the endosperm, this central cell, is developing with the 2 nuclei and the third sperm. So now it's 3N. It's going to start developing into the endosperm. And they just put an arrow there. A lot of stuff happens in that one arrow going off to the right, but eventually you'll end up with the embryo and then the endosperm surrounding that. And then the seed coat around that. So the seed coat is actually coming from the integuments. All right, so the integuments are there to protect the initial part of what the megagametophite, and protecting all of that. And then it still provides a mechanism of protection because that develops into the seed coat. And so again, it's protecting that precious embryo inside. Right, so a very nice protective function all the way through. And then the endosperm is really important because it's providing that nutrition for the baby embryo. So now, having said that, I just told you, endosperm is really important. However, sometimes what you see is a little different. By the time the seed is fully mature, you might not find an endosperm. It might have got converted into really big cotyledons. So it depends on the plant. So here, let's look at this top left. You see an endosperm tissue. You see 2 cotyledons. Both are there. Great. Both can provide nutrition. The cotyledons they call them seed leaves, but they're not like the true leaves. They're just there to kind of provide a little bit of extra support for nutrition. The endosperm is there for nutrition. Now let's look down low in the lower left. You don't see the word endosperm there in that seed. It's gone. But it was there. It just morphed into cotyledons. So the cotyledons here, all of that, can't do that. I was about to write on my laptop with a sharp marker. Maybe not such a good idea. Although I'd love to throw this through the window. My computer crashed right before lecture. I'm just like, if I don't have a laptop, I can't hold class. So it's like, what the heck? Luckily they fixed it, but it's not working. So this whole section, I don't know if you can see my reddish marks there. That whole thing is a cotyledon. There's 2 of them there. So like kidney beans, when you crack open a kidney bean, it's 2 fat cotyledons, no endosperm. But basically what happened is the endosperm kind of morphed into these cotyledons and they have a distinctive look. So they gave them that separate word. Right? So it just depends on the plant species. If you have, is it just cotyledon plus endosperm, just endosperm, what's going on? And so it just depends. And then you don't always see the cotyledons, like there's no guarantee you'll see them because it depends on if they pop up above the soil or not. Like pea cotyledons stay below ground, bean cotyledons pop up. And so here you're seeing those big fat cotyledons. Oh, they're still fresh here, but they're going to get siphoned. The nutrients are going to get siphoned off, and then they get withered and turn brown and fall. So they're just there to provide nutritional support. They get siphoned of nutrition for the real leaves that are forming. And then it just depends on the plant, whether or not those cotyledons stay down or come above ground. Yes. Yep, they get siphoned out. They're just there to provide nutrition and the early stages of nutrition. So when we look at, this idea of all these angiosperms, there's over 250,000 different species of angiosperms, and they all have some kind of flower design in order to get this all to happen from start to finish. So in order to do that, many of these plants will work with a partner in order to get pollinated. And so this is the idea of co-evolution, where you have another organism that has co-evolved with that plant and its flower over time. And it's a very specific relationship between the pollinator organism and the flower, the plant. And so they influence each other, and they still do to this day. Like we're seeing flowers shifting in their color. We're seeing them shift in some other properties, and the pollinator is able to co-evolve with that. It's kind of amazing. With climate change, we're seeing some shifts going on, and yet coevolution going on is still working. They still co-evolve with each other. It's really amazing how they're adapting to each other and evolving as partners. Yes, yes, yeah. It's amazing how adaptable through this whole process. So we're going to get into that a little bit on Wednesday and talk about fruit production too. Tomorrow in lab, you will go through fruits and have some fun with different kinds of fruits. So maybe come a little bit hungry. You might be able to eat some of that when you're done. So give that a shot.